could see the title slide up here, is Ann Moser. She's world-known, an expert in peroxisomal biogenesis defects and especially X-linked ALD. She's a research associate for neurology and co-directs the peroxisomal diseases laboratory at the Hugo Moser Research Institute. She got her bachelor's at Radcliffe and then also began working at Kennedy Krieger and has been there ever since. Her lab processes many samples from all over the world every day for PBDs. So Ann will talk about the history and diagnosis of ALD.
Thank you, Michelle, and thank you for inviting me to speak at this historic symposium on ALD. So I could not proceed with the newborn screening without the help of Walter Hubbard and Jerry Raymond. So I have to thank them so much for their participation. So what is this? Allow? Always allow? Maybe that'll get rid of it. Okay, and this is in memory of my husband, Hugo. So I'm going to describe X-linked adrenoleukodystrophy. It affects the nervous system, white matter, adrenal cortex, and testes. There's a childhood cerebral form, the adult spastic paraparesis, and Addison-only forms, which can occur in the same family, that is, with the same mutation. There is an accumulation of saturated very long-chain fatty acids due to an impaired function of the peroxisomal very long-chain fatty acid CoA ligase, and the gene is on the XQ28, codes for a peroxisomal membrane protein that is part of the ATP binding cassette transporter superfamily. There are two main forms of ALD. There's the adrenomyeloneuropathy form, which affects the spinal cord, and it affects adult males and about half of the carriers. Then there's the inflammatory cerebral form, which affects approximately 35% of males, and however, boys mainly. However, some AMN men may develop the adult cerebral demyelination. There's no correlation with mutation, and an autosomal modifier gene is suspected. The mouse model, unfortunately, has the adult AMN form, no cerebral involvement. So the history. In 1923, it was described, the childhood form was described by Zimmerling and Creutzfeldt, and it was called bronzed Schilder's disease. In 1976, the very long-chain fatty acid excess was found by Powers, Schomburg, Kuniko, Suzuki, and Igarashi at Albert Einstein in New York. 1976, AMN was recognized by the group in Vienna, and Jack Griffin, who was then at NIH. And it took us a while after we knew that there was a very long-chain fatty acid excess to discover that we could diagnose ALD by measuring the plasma total lipid very long-chain fatty acids. That was in 1981. And we were also working with Barbara Mijan at Hopkins, and through informative fibroblasts, we did some cloning of the fibroblasts and mapped the gene to XQ28. In 1990, the first successful bone marrow transplant was done for ALD by Patrick Oborg in Paris. In 1993, the gene was found by Patrick Oborg and Dr. Mandel in Strasbourg. 1997, the mouse model was produced by the group at Kennedy, Krieger, and Hopkins under the direction of Kirby Smith. So this is the identification of the abnormality in ALD. This was done by Dr. Igarashi. They knew that the cholesterol esters pathologically looked different than control cholesterol esters in the adrenal, and that was described by Jim Powers. So Dr. Igarashi isolated the cholesterol esters from brain, and the story is that he left the instrument on, and lo and behold, found this tail of very long-chain fatty acids in the ALD sample, but not in the control. And my husband wrote an article called Diagnostic Junk. You have to pay attention to the junk that you find. And these fatty acids were confirmed by Dr. Kishimoto, who was then at Kennedy Krieger, and the rest is history because we worked together with Dr. Kishimoto. So this is a picture of Kuni Suzuki, who's here, and he was also very instrumental in describing Krabbe's disease. So it's fitting now that ALD and Krabbe's are screened for together. So this is the first publication in 1981 of the plasma total lipid very long-chain fatty acids. This is before we were able to diagnose with plasma, we had to do cultured skin fibroblasts, and you probably all know how difficult it is to get a skin biopsy from boys, and families don't like it. But so this was a landmark, and then it made it easy to get diagnosis made for ALD. And then about 10 years, 18 years later, we had done 3,000 patients and 29,000 controls. So we had done a lot of samples. And back then, we were about the only lab in the US doing diagnosis. But that's changed tremendously. So with the plasma very long-chain fatty acid test, we began to screen families of the boys, and we also had the gene at this time, so we were able to find out what the incidence of ALD was. And we did that through extended family screening. And at this time, the Mayo Clinic and the Kennedy Krieger are about the only labs in the US to do the tests. So having the Mayo Clinic follow-ups and then our follow-ups, we were able to determine the minimum frequency of ALD in the United States. So males, it's 1 in 21,000. And females, it's 1 in 14,000, and if you add those together, you get a combined estimate of the hemizygotes and heterozygotes of 1 in 16,800. And the ALD was being diagnosed in France and in Canada, and they found similar results. And note that about 60% of carriers do develop symptoms in middle age or later. So, and then here's the gene publication. The ALD gene was found by Jean-Louis Mandel and Patrick Oborg in France. It maps to the XQ28. Codes for 745 amino acid and it's a member of the ATP binding cassette transporter superfamily. There are 10 exons to this gene, so it's rather large. And I went out on the website, and I found their last update was September 2nd of this year, and they had 1,585 mutations, of which 695 are non-recurrent. So you can't do mutation analysis for newborn screening. There's just too many mutations. It would just not be feasible. And there's no correlation. Genotype does not correlate with phenotype. And mutation analysis is essential for accurate heterozygote detection because the very long-chain fatty acid test in plasma has a 20% false negative rate. So you really have to identify the mutation in the family and then screen the women at risk. So the ALD phenotypes and their relative frequency, the childhood cerebral form is 30 to 40%, and the adrenoleukodystrophy form is 40 to 45%. Addison's disease is 20 to 30%. Now, Dr. Raymond will give you a lot more clinical information than I am. I'm just skimming the surface here because I'm mainly a biochemist and not a physician. The phenotypes frequently co-occur in the same family. So the childhood cerebral form is 35% of the ALD population. The onset is usually before 10 years. The earliest is 2.75 years, but it may have changed since. Peak is 7 years. Initial symptoms represent attention deficit hyperactivity, and these boys may respond to Ritalin. And so they think that this boy just has hyperactivity. However, the MRI abnormality precedes the clinical findings. So if the boy regresses and they do an MRI, they discover that it's usually too late to do any therapy. And that's the whole reason we advocate newborn screening. Here's 2 pictures of adults with the spastic paraparesis, ataxia, bladder dysfunction, muscle atrophy. They have dark skin and thin hair. And when you see a man with AMN walking, you say, oh, I've seen that before, of course. But they're rather difficult to diagnose if there's no family history. Because they may think of ALS or multiple sclerosis, and they go through all these tests. So it's also good to do newborn screening to identify these men or boys who will develop the adult form. AMN and the childhood cerebral often co-occur within the same kindred, and in fact, it's more common to have both forms. Of the 178 kindreds that we tested in the study, 50% had both the childhood form and the adult form co-occurring. So this is a slide that I took from my husband's 2002 presentation. He called it ALD Diagnosis, Future Directions. And he said, develop mass neonatal screening for males. The plasma very long-chain fatty acids are increased the day of birth. And he envisioned add tandem mass spectrometry, very long-chain fatty acid screening to existing programs. And Dr. Adrenoleukodystrophy did a study of feasibility, but his group didn't come up with a test. And then treat all ALD boys 2 or more years before symptoms occur. And he also advocated ALD heterozygote identification by mutation analysis, because we knew that we had false negatives. So the purpose of newborn screening for ALD is to prevent overt adrenal insufficiency. We know of many cases of ALD boys dying during an Addisonian crisis. They have a fever and go to the hospital, and the emergency physicians wouldn't be able to diagnose what was wrong in time, and the kid would die of Addison's disease. So, and then reduce the risk for childhood phenotype with Lorenzo's oil diet. I'm not going to get into that, but that's one of the purposes. Improve the prognosis of cerebral ALD by facilitating early bone hematopoietic stem cell and gene transplants, and 4, to improve the success of future therapies. To identify 80% of ALD heterozygotes, which would provide family screening for male ALD relatives. And then we can diagnose the disorders of peroxisome biogenesis and single enzyme defects of very long-chain fatty acid oxidation. These are very severe clinical phenotypes, and usually they present with symptoms at birth and they end up in the NICUs. So we can help prevent the diagnostic odyssey of these infants and provide genetic counseling and early supportive therapy. I would have to have a whole nother lecture to describe the disorders that can be, to describe the peroxisome biogenesis disorders, but they also have a defect in very long-chain fatty acid oxidation. ALD is the most common peroxisomal disorder, and secondarily, the peroxisome biogenesis disorders are the second most common. So here's the history of the development of the newborn screening test for ALD. There was a 2004 meeting at NIH on newborn screening that I attended, and ALD, I saw, was added to the list of disorders recommended for screening. But it was in the category that there was no test available on the newborn blood spot, so it wasn't recommended at that time. In 2006, the LCMSMS assay for ALD newborn screening was published by Walter Hubbard and our group. And then we did those analyses without having a real internal standard, nor did we have the C26 lyso-PC. These were custom synthesized by Avanti for us, and at first they made the D9 derivative, which wasn't very pure, and then they made a D4 derivative, which is very pure. In 2009, we validated the tandem-spec assay for ALD newborn screening. And then we made an attempt to combine the acylcarnitine with the C26 lyso-PC screening by LCMSMS. It worked very well, but most people are happy with the way they do acylcarnitines and don't want to add ALD to that assay, so it's not being used. And then Chris Haynes at the CDC developed a negative mode LCMSMS for ALD newborn screening that was more specific, but it's less sensitive. And then in 2013, we published our results of the Maryland newborn screening pilot of 5,000 consented newborns were screened here. Now, this is a plot of the plasma total lipid very long chain fatty acid, C26. And the test is done on black dots or ALD babies. And we showed that even at the day of birth, the C26 level is increased. And that's published, okay? And that was published in 1999. And then we went and took a look at whole blood spots that we had saved from our ALD patients. And at first, we just took an 1/8-inch punch and treated it with acid and got the total lipid C26, 24 levels. However, we did not expect this to work because of the fact that the red cells are present. And when we test the plasma very long-chain fatty acids, we have a false positive with hemolyzed samples. So we knew that red cells interfered with that assay. So we expected that this would not be a test for newborn screening. Then Rizzo et al. Published that they did an extensive look at the acylcarnitine profile in ALD and found that it was normal, so you can't use the acylcarnitine for diagnosis. Then together with Walter Hubbard, I measured all the species of sphingomyelin and ceramide by LCMSMS. And there was an increase in the C26 and 24, but there was an overlap with normal. So we couldn't use those lipids for diagnosis. Now, I knew from our work in brain with Dr. Christiana Tata, when she was in our lab, we isolated lipids from the apparently normal white matter of an ALD brain, and we looked at the lipid and found that the phospholipids in the normal appearing white matter had the greatest amount of C26 when compared to control myelin. So I said, let's look at phospholipids. But as you know, if you've done lipid LCMSMS, it's rather complicated to look at phosphatidylcholine because there are 2 fatty acids. So you have a lot of species to look at. So it was very complicated. So Walter Hubbard and I put our heads together and we said we should look at the lysophosphatidylcholine, where you take off the fatty acid on the SN2 position, which is usually a polyunsaturated fatty acid anyway. So you're left with the SN1 position, which is usually a saturated fatty acid or a monounsaturated fatty acid. So we look at the lyso-PCs, and Walter Hubbard had an off-the-shelf method because he had been looking at lyso-PCs and breath exudates of asthmatics. Oops. So lo and behold, there was a 10-fold increase in the C26 lyso-PC in ALD dry blood spots when compared with control. So we had a test, and it was first published in 2006. So these are the lipids we're looking at. Where's my mouse? This is the one that we're talking about. This is the lyso-PC and there would be 26 carbon fatty acid here. So, and then I also had looked at ceramide and sphingomyelin, which are sphingolipids, and they're very rich in the saturated, very long-chain fatty acids, but not diagnostic. And here we are with the results from the paper showing a 10-fold increase in the C26 content of ALD and AMN versus controls. And the peroxisome biogenesis disorders have even higher amounts. So it's a very good test. And then we compared our normal controls with 30 neonatal blood spots, and we found about the same results for the C26 lyso-PC. So this is the paper. Now, after this paper came out, we had custom synthesis by Avanti, and this is the C26 lyso-PC with the 26 carbon fatty acid here. And then they made a successful deuterated internal standard with the 4 deuteriums added to the fatty acid. And then you can either use, for MRMs, you can use the 184 or the 104. However, Sylvia Tortorelli pointed out, we had used the 184 initially, but it was rather, there were a lot of isobaric compounds. So she recommended, and we followed her advice, that 104 was a better MRM. Less sensitivity, but it's more specific. So here's the method we currently use, and we used for the newborn screening of the 5,000. We just sample with an 1/8-inch punch into 96 well plates. We add 150 methanol containing 10.6 picomoles of deuterium labeled internal standard. Cover the plate with to prevent evaporation, and mix gently for 1 hour at room temp. Centrifuge and transfer the soup to a clean 96 well plate. And on the API 4000 instrument, we inject 2 microliters, but for the 3200, which is less sensitive, we use 10 microliters. And then I have, you can do a long procedure, which is 7 minutes, which is a 4.6 by 50 millimeter column, or you can use a smaller bore column and do a 2-minute isocratic run, which takes less than 2 minutes, actually. And this is the isocratic run using a narrow bore column on the API 4000. And here's a control dry blood spot. This is the C26 lyso-PC peak. And you can see we have to separate it from this isobaric compound. And we can only get rid of that by doing negative ion, but we prefer chromatography in positive ion mode, so we live with this isobaric peak. And so here is a positive sample showing the increased amount of C26 lyso-PC. So that's the only chromatography you're going to see. Now here are the bar graphs for comparing the venous blood spots with the newborn blood spots. So if it doesn't have any NBS next to it, it's a venous blood spot. So done 190 normals in this graph. Here are the newborn blood spots. They're a little bit higher than the overall than the venous blood spots. And here are the 18 ALD and PBD newborn blood spots that we retrieved from the state of California and the state of Michigan. We knew families, ALD families and PBD families from both those states, and we were able to retrieve the blood spots for these analyses. And here are ALD venous blood spots showing that they're lower on mean than the newborn. And here are the heterozygotes, clearly higher than the controls, but there is some overlap with controls because we can only detect about 80% of the females. And I did these studies with Dr. Steinberg, who was getting whole blood for mutation analysis from women. And so I was able to determine that they were normal by mutation analysis or they were carrier by mutation analysis. So these are very accurate diagnosed ALD heterozygotes. And here are the peroxisome biogenesis disorders, which on average are higher than the ALD venous blood samples. So you can also use the C24 lyso-PC for diagnosis. However, I look at it if I'm questioning the C26 levels, but I think the absolute levels of C26 are probably the best measurement, and I don't often use the C24. I mean, we measure it, but I don't often look at the results that closely. And here's the summary that was published in the paper, showing that there's a clear difference between the normal newborns and the ALD and PBD newborns. And this is just looking at the C26 lyso-PC level as picomoles per 1/8 inch blood spot. The reason I have 16 here and I had 18 before is that I was able to get 2 more samples in follow-up from our prenatal diagnoses from families that elected to keep their ALD baby. So here's the publication in 2009, the validation of the combined liquid chromatography tandem mass spectrometry method. And a lot of people participated in this study. And I thank them all. So the C26 lyso-PC newborn screening test identifies the following disorders: X-linked adrenoleukodystrophy, which we predict affects 1 in 16,800 births. And it has these 3 phenotypes: a childhood cerebral phenotype, the adrenomyeloneuropathy, and Addison's only. The test identifies 80% of ALD heterozygotes, we predict based on our analyses. And it will also diagnose the peroxisomal biogenesis disorders and the single enzyme defects of peroxisomal fatty acid oxidation. And this is the paper on the combined extraction of acylcarnitines and C26 lysophosphatidylcholine from dried blood spots. And then, together with the Maryland State Screening Lab, we performed a prospective study to offer optional testing for ALD and PBD to parents of 5,000 newborns born in 3 hospitals in the Baltimore area. We've finished analyzing the consented newborn samples with no positives. And we hope to start ALD newborn screening in Maryland after the Maryland State Screening Lab moves to their new facility. They haven't done so yet. We keep waiting for them to move. And then we hope to start in January 2015, and if they don't, I'm going to sit on their doorstep every day because they'll be 2 doors down from me. So this is the publication for the newborn screening that we did in Maryland. So I wanted to say a little word about the importance of the ALD families and all the support groups. They have been magnificent in their lobbying for ALD newborn screening. And as you know, lobbying in New York has resulted in New York's adding it to their panel of tests. New Jersey, Connecticut, Maryland, and California, we all have legislative approval to add ALD newborn screening. And as far as I know, the lobbying continues in Texas, Illinois, Florida, and Massachusetts. So this is the cast of characters. Without, we wouldn't be here today speaking about ALD newborn screening. And we had funding from a lot of family support groups. We had wonderful collaborators in the state of Maryland and state of California, the Mayo Clinic, and Baltimore hospitals. That's it. If you have any questions, I'm here throughout the whole meeting.